and just hello everyone, welcome to class 12 biology. In this video we will discuss about genetic disorders, and genetic disorders are of two types: Mendelian disorders and chromosomal disorders. So first we will try to know the difference between these two disorders, Mendelian disorders and chromosomal disorders. So you can write this table down in your notebooks. We can see the differences between the two disorders, genetic disorders. So the first one is Mendelian disorders. The Mendelian disorders are determined by alterations or mutations in a single gene. If there's a mutation in a single gene, this will lead to Mendelian disorders. Now this is called as Mendelian disorders because the transmission or the inheritance pattern follows the principle of inheritance which was proposed by Gregor Mendel. So therefore it is called as Mendelian disorders. So the Mendelian disorders can be either dominant or it can be recessive. And it could be located on autosomes or it could be located on sex chromosomes. So therefore they could be autosomal or sex-linked. And since this is following the Mendelian pattern, it can be traced by pedigree analysis. So it can be easily traced within the family by pedigree analysis. Whereas chromosomal disorders, they are caused due to absence or excess of one or more chromosomes. If an organism has got an extra chromosome or one less chromosome or more than one chromosome. So such kind of situations could lead to some disorders or diseases which are called as chromosomal disorders. So we will see some examples later on. Failure of segregation. So how is this caused? It is caused, what causes a gain of extra chromosome or loss of extra chromosome. Loss of chromosome is because of the failure of segregation of chromatids. If the chromatids, they fail to segregate during the anaphase, so what happens is one of the two cells will get one extra chromosome. Or it can also be caused due to failure of cytokinesis after telophase. If the cell fails to divide, if there is no cytokinesis, if the cytoplasm does not divide after telophase, so this can lead to a loss or gain of chromosomes. An entire set of chromosomes can be lost or gained. So these are the two reasons which can lead to chromosomal disorders. So now I have given you the differences between Mendelian disorders and chromosomal disorders. Now, in order to understand this Mendelian disorders, the cause is mutation, right? Mendelian disorders. So it is caused by mutation in a single gene. So let's try to understand what is mutation, right? So mutation, in order to understand mutation, now we need to understand what is gene. We need to be clear about the concept of gene. So genes, they are said to be sequence of DNA. Gene, it is a sequence of DNA which codes for a particular product, and that product can be either protein or RNA. So the DNA, which is a polynucleotide, a short sequence of DNA which can code for a particular protein or an RNA is called as a gene. Okay, so this gene, it determines the phenotype of an organism. So the phenotype of an organism is determined by that particular sequence of gene. So our eyes, our hair color can be black because there's a short segment of DNA which codes for a particular protein which makes our hair black. So that's how the concept flows. That's gene. So now what is mutation of the gene? So what causes the mutation of the gene? Mutation is a phenomenon which results in the alteration of DNA sequence. So the DNA is a sequence of nucleotides, right? It's a polynucleotide. So if that sequence is somewhat altered, right, which can result in the formation or which can result in some kind of abnormalities in genotype or phenotype of an organism, that is called as mutation. So what causes mutations will be mutagens. So there are mutagens, physical mutagens and chemical mutagens. Mutagens are nothing but those factors which can cause the alteration of the DNA sequence. So those are mutagens. So I have given you a picture over here. You can observe this one. Let's see, this is a short segment of DNA given over here. So let's say this short of segment of DNA is the normal gene, and this normal gene codes for a normal protein. So this protein will function and produce a normal phenotype. So this is a normal gene and its result. Okay, so this will result to a normal protein and normal phenotype. However, if there is some kind of alteration, you can see over here one strand is broken over here. So this is a mutated gene. So let's say it has undergone a mutation, right? So this is a mutated gene. So mutated gene, if the gene gets mutated, what can happen is it can either produce an abnormal protein or it won't be producing any protein at all. So what happens is if a mutation happens, it will affect the protein, and the protein, if the protein is affected, the phenotype will be affected. So the normal gene will produce a normal protein, will lead to normal phenotype. However, mutated gene will result in abnormal protein or no protein, and this will lead to an abnormal phenotype. So that is the mutation concept, right? So let's try to understand some forms of mutation, chromosomal aberrations. So the first one is chromosomal aberrations. Chromosomal aberrations, we are studying this because we know that DNA or chromosomes are formed by DNA, right? So if there is a loss or gain of DNA segments, that will lead to chromosomal aberrations. So let's see over here, this first one. So the chromosomal aberrations are of these three types. There are actually more than that. So let's discuss about these three first, right? The chromosomal aberrations are of these three types: deletion, duplication, and inversion. Now, if you look at the pictures carefully, you can see that in the first part, the deletion part, what happens is this particular segment D, it gets deleted. It gets removed somehow, right? So when this gets deleted, you can see over here A, B, C, and D is missing. It forms E, F, G, H. So a short segment of the chromosome is removed or deleted. So that is called as deletion, and this can lead to some kind of disorder. Over here in duplication, what happens is you can look at this section B and C. This section B and C, it gets duplicated. Duplicated means it forms a extra copy of itself. So in this chromosome, there is increase in the chromosomal length. So this is called as duplication. So what happens in inversion? Look at it, inversion. So if you look at B, C, D, these three sections, what happens is the sequence, it is reversed. So this is called as inversion. So these are the three forms of chromosomal aberration. So you can try to understand how chromosomal aberrations can happen. It can happen due to some kind of physical or chemical factors, or it can also be formed due to some mistakes that happens during DNA replication. The next is point mutation. So the first one was chromosomal aberrations. Now the second one is point mutations. So what are point mutations? Point mutations are the mutation due to change in a single base pair of a DNA. So the DNA is a polynucleotide, and polynucleotides are formed by a base, a sugar, and a phosphate group. So this we will study later on. Don't worry about this right now. So let's see. So DNA, it has got bases, and bases, they form base pairs. Now mutation which happens due to a change in a single base pair is called as point mutation. So the point mutation are of these three types. You have to remember that. So this is the normal gene, right? This is a normal gene in which you can see the sequence of base pairs over here, right? In this one, you can see the sequence of base pairs. And this gene will lead to formation of a protein or a protein, right? So this is a sequence of amino acids over here, polypeptide. Okay, so this is a polypeptide or protein. You can see the individual amino acids over here. So the normal gene will produce this sequence of amino acids. Now let's look at each form of point mutation. So this is the first form of point mutation, substitution. So in substitution, what happens is one base gets substituted by some other base. So over here, you can see this. In the normal one, we have GCA. Over here, we have GTA. So that means the C is substituted by T, right? The C is kicked out and T is substituted over here. So this leads to substitution mutation. So in substitution mutation, what happens is, you can see over here, in normal case, methionine is followed by arginine. But in substituted or the mutated gene, right, it leads to the formation of methionine followed by histone, right, histine. So that amino acid sequence is changed over here. So such amino acid sequence change can also lead to a different phenotype. So that is first form of point mutation. The second form of point mutation is insertion. So the word itself is self-explanatory again. There is an insertion or addition of one base over here. So TAC, it is followed by G, right? But over here you can see that between TAC and GCA there is insertion of an adenine over here. So when adenine is added over here, what happens is the entire amino acid sequence gets changed over here, because the genes are read in triplets. So this we will study later on, I'm telling you again. So here you can see that insertion of a base can lead to different polypeptide. So this will lead to a different phenotype. And the third one is deletion. So it is an opposite of insertion. What happens is the G, right, the base G is removed. When the base G is removed, what happens? Again, the amino acid sequence gets changed, and this will affect the phenotype. So these are the forms of mutations, point mutations over here. We have studied, now we have discussed about chromosomal aberrations. Chromosomal aberrations are of these three types: deletion, duplication, and inversion. And point mutations. And point mutations are of these three types: substitution, insertion, and deletion. Now we can start the Mendelian disorders, right? Some examples of Mendelian disorders. So the first Mendelian disorder example provided in your textbook is color blindness. So color blindness, it is said to be sex-linked recessive disorder. It is said to be sex-linked because the gene responsible for color blindness is located on X chromosome. So this defective gene, what it does is it leads to formation of defective cones within your eyes. And defect in cones leads to failure to discriminate between colors, and mostly between red and green. So a person suffering from color blindness won't be able to differentiate between the shades of red and shades of green. And if you perform an online test by yourself, you will be able to come across such pictures, and you will be able to diagnose yourself whether you're suffering from color blindness or not. If you are suffering from color blindness, you won't be able to tell what numbers are hidden within these pictures. Now the peculiar thing about this disease is that 8% of males, they suffer from color blindness, and only 0.4% of females, they suffer from color blindness. So why there is less number of females suffering from color blindness? The simple answer is because the gene responsible for color blindness is located on X chromosome, and since females, they have got two X chromosomes, and since the disease is a recessive disorder, if one of the X chromosomes of the female carries normal gene, she won't be suffering from color blindness. However, the males, they have got only one X chromosome, and if that lone chromosome, X chromosome, carries the defective gene, he will be suffering from color blindness. And this will become much clearer if you go through this particular cross between these two individuals. We have one normal father. The father is not suffering from color blindness, and we have a carrier mother. So this is said to be carrier because she is not suffering from color blindness, but she is carrying the color blindness gene on one of her X chromosomes. Now, if you look at the possibilities of their offsprings, genotypic possibilities of their offsprings, we can see that there are 3 individuals who do not suffer from color blindness, right? And out of these 3 individuals, 2 are girls. So the girls in this particular family will never suffer from color blindness. One girl will be fully normal, whereas one girl will be normal, but she will be carrier. And this boy will be fully normal. But look at here, out of these 2 boys, one boy will be suffering from color blindness, because if he receives this X chromosome from the mother, the X chromosome which is carrying color blindness gene from the mother, he is bound to suffer from color blindness. So therefore, there are more number of boys who suffer from color blindness compared to girls, because girls, they have got 2 X chromosomes. If one of the X chromosomes is normal, then she won't be suffering from color blindness, but she will be a carrier. So that is about color blindness. So the second example of Mendelian disorder is hemophilia. Hemophilia is a sex-linked recessive disease in which a protein which plays an important role in blood clotting mechanism is affected. A simple cut will lead to death due to non-stop bleeding in such patients. So a person who is suffering from hemophilia, he will have a defective gene, and that defective gene won't be able to produce a protein which plays an important role in blood clotting mechanism. So when he or she receives a simple cut, he or she will die due to non-stop bleeding. So that is about hemophilia. Now there's one important question regarding hemophilia which has been constantly asked in board examinations. That is, why possibility of a female becoming a hemophilic is extremely rare. Why is it so rare to find a female who is suffering from hemophilia? Now the answer is, for a female to suffer from hemophilia, she should have both her X chromosomes with the defective gene. And that can only happen when mother of such a female is a carrier and the father is a hemophilic. But the second criteria is improbable, because the father won't be able to reach his reproductive age. So unviable in later stages of life. That's what it is written over here, right? If you look into your textbook, you will find this particular statement given. So what it means is, a female to suffer from hemophilia, she has to have a father who is hemophilic, and that is not possible because males who suffer from hemophilia are unable to reach reproductive age. So that is the answer. So the third example is sickle cell anemia. Sickle cell anemia is an autosomal recessive disease. Remember, the first two examples were sex-linked recessive disease. Now this one is autosomal recessive disease because the gene responsible for this particular disease is located on an autosome and not on sex chromosome. So in this disease, what we have is a person suffering from sickle cell anemia will have sickle-shaped RBCs. So that's what makes a person suffer from sickle cell anemia. And this disease is controlled by HBA and HBS alleles. So these are the two alleles in which the HBA allele will lead to the formation of normal RBC shape, that is biconcave shape, and HBS allele leads to formation of sickle shaped RBC. Since this is a recessive disease, only those individuals who have got homozygous recessive genotype will suffer from the disease, whereas the other people, they won't suffer from the disease. So what leads to the formation of sickle shaped cell? So this picture is given in your textbook. You should observe this particular picture carefully, and then you have to be able to explain what is happening over here. So this is a normal gene. So this is HBA gene. So this is a normal gene which leads to formation of a normal RBC shape, in which what we have is, we have this particular gene leads to formation of an mRNA, GAG, and it will lead to formation of glutamine in this particular amino acid sequence. So look at the sixth amino acid, it is glutamic acid. Right. Now, a person who is carrying HBS gene, right, the sickle cell gene, what has happened over here is, there has been a point mutation, right? There has a point mutation. It has a point mutation in which A, adenine, is substituted by thymine. A is substituted by T, and this substitution leads to the formation of a different mRNA sequence. A different mRNA sequence will lead to formation of a different amino acid on the sixth location. Now, in normal hemoglobin, what we have is, we have glutamine. Now, in this sickle cell, a person who is carrying sickle cell gene, it is carrying valine. So this point mutation, which leads to the formation of the change in one amino acid in the sequence, leads to the formation of sickle shaped cell. Right. So that is an effect of point mutation, and it leads to sickle cell anemia. So fourth example of Mendelian disorder is thalassemia. Thalassemia is autosomal recessive disease. In this particular disease, there is formation of abnormal hemoglobin, which leads to anemia. Abnormal hemoglobin is formed due to less synthesis of one of globin chains, alpha or beta chains. I have provided you a picture over here. This is a molecular structure of hemoglobin. You can clearly see that there are 2 alpha globin chains and 2 beta globin chains. Less synthesis of these globin chains will lead to thalassemia. Now these globin chains are proteins, and the synthesis of these proteins are controlled by genes. Right? So now, based upon alpha and beta globin chains, which one is synthesized less in amount, there are 2 types of thalassemia: alpha thalassemia and beta thalassemia. If an individual is producing less amount of alpha globin chains, that person will be suffering from alpha thalassemia. And if a person is producing less beta globin chains, then that person will be suffering from beta thalassemia. Now, thalassemia is of 2 types, alpha thalassemia and beta thalassemia. Alpha thalassemia, the disease is controlled by 2 genes which are tightly linked on chromosome number 16. That is HBA1 and HBA2. So these 2 genes, they are located tightly linked on chromosome number 16. Whereas beta thalassemia, it is controlled by only 1 gene, that is HBB, and that particular gene is located on chromosome number 11. Now you can see that alpha thalassemia genes are located on chromosome number 16 and beta thalassemia gene is located on chromosome number 11. Now again, over here I have given you the picture of these 2 genes over here. So let's say this is chromosome number 16. We have a homologous pair over here. Here we have these 2 genes, HBA1 and HBA2. They are linked tightly on chromosome number 16. If one of these genes, right, HBA1 and HBA2, if one of these genes is mutated, what will happen is there will be less or reduced synthesis of alpha globin chain. So here this person will be suffering from alpha thalassemia. Now in the second picture you can see that in this individual HBA1 is mutated. Along with that, HBA2 is also mutated. So there are two alleles are mutated. So what happens is this person will suffer from a severe version of alpha thalassemia. So now you should understand that alpha thalassemia, the mechanism with which the gene controls is quantitative in nature. If more alleles are mutated, the severity of the thalassemia will be more, right? If all these 4 alleles are mutated, what will happen? That person will be suffering from the most severe form of thalassemia, because there will be a severe reduction in the production of alpha globin chains. So that is why this effect is said to be quantitative. So that is about thalassemia. So now next we will talk about chromosomal disorders. Chromosomal disorders are caused due to absence or excess of 1 or more chromosomes. So there are 2 types of chromosomal disorders. First one is aneuploidy, and then second one is polyploidy. Aneuploidy is gain or loss of a chromosome. A gain or a loss of a chromosome is called as aneuploidy, whereas increase in a whole set of chromosome is called as polyploidy. So in order to understand this, let's look at human karyotype. Over here we have got the human male karyotype, normal human male karyotype, and this is a normal female human karyotype. We know that we have got 23 pairs of chromosomes. We have got 22 pairs of autosomes and 1 pair of sex chromosomes in both male and female. So now this is the normal karyotype. This is what a normal human being will have, right? But if a person gains or loses 1 chromosome somewhere over here, right? Let's say if he gets 1 chromosome over here, then that person is having aneuploidy. Whereas if a person gains an entire set of these chromosomes in a cell, then that person is suffering from polyploidy. But polyploidy is very rare in animals. It is more common in plants. That's what it is said, right? Polyploidy is more common in plants and less common in animals. In animals, polyploidy will lead to abortion. Now, the first example for chromosomal disorder is Down syndrome, and this is also called as trisomy 21 because if you look at the karyotype over here, this is a karyotype of a person suffering from Down syndrome. Now you have to remember the normal karyotype over here, right? There is a pair of chromosome on all the numbers over here, right? All 22 chromosomes, they are in pairs, and the last one is the pair of sex chromosomes, right? So if you look at Down syndrome, a person suffering from Down syndrome, if you look at that person's karyotype, you can see over here that at chromosome number 21, there is one extra chromosome. So there is one extra chromosome number 21 over here. So therefore, this is also called as trisomy 21. So a person suffering from Down syndrome will show all these symptoms, right, or characters. So that person will be short-statured, they will have small round head, they will have furrowed tongue, they will have partially open mouth, the palm will be broad with characteristic palm crease. Over here you can see that particular crease, palm crease, and there will be physical, psychomotor and mental retardation in person suffering from Down syndrome. So the second chromosomal disorder is Klinefelter syndrome. In Klinefelter syndrome, we have got an extra X chromosome, right? There is an extra X chromosome over here. You can look over here at the sex chromosome side. You can see that there is an extra X chromosome. That is why it is also called as XXY. So a person who has got extra X chromosome, that person is said to have all these characters. But in our textbook, it is given that the person will be overall masculine. That is, the person will be born a male, but that person will show certain kind of feminine characters like gynecomastia, which means development of breast, and that person will be sterile, right? Sterile means he won't be able to reproduce. So that is Klinefelter syndrome. And the last one provided for chromosomal disorder is Turner syndrome. Turner syndrome is said to be XO, right? It is said to be XO because you look over here, there is one X chromosome missing. Look at the karyotype of a person suffering from Turner syndrome. You can see that one X chromosome is missing. So that is why it is called as XO. A person who has got Turner syndrome is said to have all these characteristics. But in our textbook, what is there? It is said that they are born as females. These individuals, a person suffering from Turner syndromes, they are born females, but they are sterile females. Their ovaries won't be functioning. They are sterile females, and they lack secondary sexual characters, right? They lack secondary sexual characters. And then you can also look at other symptoms as well. So that is Turner syndrome. So that is all about disorders, genetic disorders. So genetic disorders, I said, are of two types. What are they? Mendelian disorders and chromosomal disorders. And in Mendelian disorders, I have given you 4 examples. There is also a 5th example, phenylketonuria. I have not discussed about that because we have already learned about phenylketonuria in our previous videos. So, and then there are chromosomal disorders. I have given you 3 examples for chromosomal disorders: Down syndrome, Turner syndrome, and Klinefelter syndrome. So with this, we have come to the end of the chapter number 5, and in the next video we will start with chapter number 6, that is molecular basis of inheritance, in which we will study about DNA and in detail about how DNA acts as a genetic material. Thank you.